Department of Reproductive Biology in School of Medicine, and I'm filling in for Dr. Weaver. This lecture and a couple of others on reproduction. So this lecture is on signaling. So how does one cell talk to another cell? Okay? How do chemical signals in the body actually do what they do? And so this is straight out of the notes that Tim has in the Canvas. So I'm just going to essentially go by his notes and try and fill in anything that needs to be filled in. Ask questions if you have them. And so this is sort of the fundamentals of endocrinology in a way. And as dentists, I know you're more interested in what's going on in the mouth rather than what's going on with the thyroid gland, but it does all relate. You need to have some type of a foundation on physiology in general in order to be a good clinician overall. And so this is important for anything that you do in the biomedical business. Okay? So, as I said, what we're going to talk about is how signals are transmitted from one cell to another, and how the body in general is able to maintain homeostasis. And it really is homeostasis this is high school biology. You all heard the word homeostasis, but that's what keeps us normal. And as soon as we start deviating from homeostasis, that's where pathology comes in. And in order to maintain homeostasis, cells need to know what to do and when to do it. Okay? So, as is shown up here, at least half the slide, the screen, cells need to survive. Right? So that's the first thing a cell needs to do is live and do whatever it needs to do to maintain its intracellular homeostasis. It needs to sense stuff, so what's going on in the environment around the cell, and then it needs to be able to somehow respond to what's going on around itself in a physiological manner. So it just doesn't respond haphazardly. We've evolved so that cells will respond to each other and to the environment they're in, such that as an organism, we can survive. So we're a multicellular organism. And so there's intermediary metabolism, and that's really what the cell is doing. It's basically taking in resources, producing energy, and then using that energy to actually be a cell. And you could argue that, so why would a cell want to be a cell? So, why is there this drive for the cell to survive? And you could argue that the whole reason that the cell is, I'm getting philosophical now, the reason that the cell is there is to keep DNA in check and under control, so that DNA can then be transferred to the next generation. So ultimately, from a single cell organism that is dividing, it's basically replicating its DNA and putting that into the next generation. Now, there's only two cells, one cell in the body that does that in us, and what's that? What cell is that? It's the gamete. Okay, so in our gonads, we have cells that will actually take DNA to the next generation. But all our other cells in our body don't do that. It's not like a cell in my finger here. That DNA is going to be transmitted to the next generation. It's not. But the DNA in all of the cells in our body is sort of, it's programmed to have that DNA in our gonads get into the next generation. So really our whole physiology is based on reproduction. Okay? And you can break that down into the single cell level. That everything we do on this planet is basically to move our DNA from one generation to the next. Okay? It sounds a little crazy, or in an evolutionary sense, that's really what we're here for. And there's a lot of stuff that goes on in between. We have fun doing it, or not. So the intermediary metabolism that the cell has to deal with evolved so that the overall physiology of the organism can be sustained. Okay? So when we look at number 2 and 3, to sense and respond, They deal with how a cell alters its behaviour to allow adaptation to changing situations. So, all right, so that's the core. Everything we do in biology, whether or not you're a dentist or a nurse or a medical student, it all comes down to evolution. Natural selection is why we're here, and it's why our bodies do what they do. Why do we have incisors? That was an adaptation why don't we have incisors the size of some other animals on the planet? Adaptation and evolution of what really brings us to where we are today. And so in order for that to happen, a cell has to do stuff. And so the ability of the cell to respond to its environment is critical. And the way we do that holistically in terms of reproduction is that our genes get mixed up during the process of reproduction such that we have meiosis happening in our gonads so that there's variability. Each one of us in this room are not clones of our parents. Okay? If we were dividing the way bacteria divides, we all look alike. But we don't. We have this variability. This variability makes every cell in our body, my body and your body, slightly different. And that slight difference allows us to respond to the environment and evolve. Okay? So the way that a cell responds to its environment is critical. And that's all dictated by the program that's in the DNA. All right? And then 3, the response. So first of all, the cell has to sense it, and 3, the cell has to respond. And the way a cell responds, as Tim says here, it depends on what proteins are made in the cell, and those proteins are made based on gene expression. So genes encode for proteins, and proteins do stuff. Okay? So whatever the cell is going to do will depend on the activity of specific proteins. Okay. So what we're going to focus on in this class is how a cell will sense things. Most of this involves communication between cells, and that involves signaling systems. So how does a cell in one part of the body signal to another cell in another part of the body, or a cell right next to it? So proteins do work. So let's begin with the idea that for anything a cell does, a protein is doing it. Any response by a cell is simply a change in total activity for one or more proteins. So proteins are sitting in each one of our cells. They're active. They're either structural proteins or they're enzymes or they're signaling molecules, but they're doing stuff. Okay? And there are certain proteins that will catalyze other reactions. And they're the proteins that we're going to be talking about today. The proteins that actually are invoked, are activated when a cell is responding to another cell or to a signal that's coming from another cell. And so the total activity of a protein can be expressed as what is the specific activity of the protein, what is it actually doing by the amount of that protein. So if you think of an enzyme in the body that converts substrate A to product B, how much of that enzyme is around and how much of that, what is the activity of that enzymatic process. So signaling systems must change the total activity of a certain protein either by altering the specific activity of existing protein molecules or by altering gene expression to change the number of molecules of that protein. Alright, so what that's basically saying is you can change the activity of a protein by switching on or off a gene that's encoding for that protein, and that changes the level. Or, importantly, in what we're going to talk about later, is that you can change the activity of the protein by changing its structure. Post-translational modification. After the gene is expressed, makes messenger RNA, messenger RNA encodes for protein. That protein is sitting around in the cell. Its activity can be changed by changing the structure of the protein. Usually that's done by phosphorylation, where either a tyrosine or a serine residue in that protein is phosphorylated. And that can switch on or off the activity of that protein. For instance, if it's an enzyme, you can switch on or off that enzyme activity. So and this adds to the complexity of the whole system itself. it's not just the number of genes that we have that are encoding proteins in each of our cells, but then it's the activated or unactivated state of each of those proteins. So the level of complexity goes up, and it's increased significantly by this process of post-translational modification. So the term signalling implies that information is being transferred from one place to another, and that it brings about a response by changing protein total activity in that target cell. So what are some examples of signalling in our body? We can divide signals into groups. So the most direct and rapid signal is direct electrical communication using gap junctions between cells. Okay? So do you all understand what gap junctions are? So when one cell is sitting next to another cell, the plasma membranes in the cells actually form this core. There's a connection, a physical connection between these two cells called a gap junction. And through that junction, chemical signaling molecules can be passed from one cell to another. Now, can anybody think where this might be important? So in the heart. So you have to coordinate the contraction of all those cardiac cells so that you get a nice pulsatile contraction. Okay? And in order to do that, there's an electrical signal that happens first. But then once that electrical signal to the ventricles is initiated, then the rest of the signaling up the ventricle is conducted by gap junctions. Can anybody think of another place in physiology where that's important? We all went through it. We were all born. Okay? Well, most of us were born in the natural way. Some of us may have been born by C-section. But if you were born by vaginal delivery, the uterus has to contract in order to push the baby out. Okay? And that contraction, that really forceful rhythmic contraction of labour that happens at birth, is facilitated by the formation of gap junctions between those muscle cells of the uterus. Okay? So gap junctions can be really important because they help the system, the organisation of those cells to form a syncytium, an organised group of cells that will behave together via this direct signalling, this direct coupling between the cells. So the direct electrical communication using gap junctions between the cells, this is the fastest form of communication in the body because they're right next to each other. All right? So what happens in one cell spills over into the neighbour. It's seen in cardiac muscle and smooth muscle. Some neurons, you'll learn more about this in the neurophysiology. Okay? It's fast, but there's not a lot of flexibility. As one cell behaves like all the other cells that it's connected to. So it's an interesting way of communication, and what we know now is that these gap junctions that form can also be regulated by phosphorylation. So they can be switched on and off, or they can be selective. For instance, if you have a gap junction between two smooth muscle cells, for instance, in the uterus, those gap junctions can let signaling molecules pass that cause relaxation, or they can let signaling molecules pass that cause contraction. Okay? So relaxation during most of the pregnancy and contraction at the end of labour can be regulated by these gap junctions. And that's usually done by phosphorylating the proteins that form that core connexin, it's called that channel. Okay? And so here again, post-translational modification of the protein by phosphorylation can affect the function of that protein. And in this case, it can affect the function of the gap junction. You can switch it on or off, or it can make it qualitatively different by selectively letting it pass through different compounds that will have a different physiological effect. So it's an important way of communication, but it's also an example of how signalling happens and is controlled. And it's critical that these signalling molecules are controlled in the context of physiology. So you don't want the uterus to contract for 28 weeks, you want it to contract for 40 weeks. So there are other processes, other elements of the system in play that keep these gap junctions in the closed or relaxatory mode most of the pregnancy. And you don't want them shutting off in your heart. Okay? It would be not a good thing if these gap junctions fail in our ticker. We would be done for. All right? So it's all chemical signaling. Right? The stuff that's passing through the gap junctions, the stuff that the cell is responding to, they're all chemicals. So this is all done by chemical signaling. So the chemical signals, they're slower than the electrical signals that happen in the nervous system and slower than gap junction mediated signals, but more flexible. All right? So now I'm talking about signals that are in the extracellular space of the cell or in the blood. So signals that are made, that are secreted, hormones that are secreted by one cell that can act on another. Now last week you learnt about endocrine, paracrine, and autocrine signaling, the way hormones are transported from one cell to another. If they're transported through the blood, that's called an endocrine signal. They can move from one cell through the extracellular space to another cell. That's a paracrine signal. And sometimes they can be secreted by a cell and act directly on the cell that's secreted. That's an autocrine signal. Okay? So when I think of hormones, I don't just think of the hormones that are in the blood that's circulating. I think of a hormone as being that secreted chemical signal that's being made by one cell that can act on another cell, or it can act on the cell that's secreted. So if we think of hormones in that way, then there are three main types of these hormonal chemical signals. There's neurotransmitters, so these are chemical messages, hormones, neurohormones, that are made at the synaptic cleft by one cell crosses a synaptic cleft, and it causes the target cell to initiate or a response, usually an action potential. So neurotransmitters, these are called. They're fast, they have short duration and short response. And they're cleared very quickly. Okay? So you don't want these signals to last too long, because you want to get in, do their thing, get out, and then once they've done their thing, get rid of them. Right? So if you think of signaling theory, anybody here an engineer? Electrical engineer? No. But in signaling theory, if I was to send a signal from here to that building, and it was an SOS, right? And I left the S on for too long. You know, you have to. Signaling is all in the context of how long that signal is there. Okay. So if I'm in a situation here where there's trouble and I have to send a distress signal over there. During the time when there's trouble, I want to send that signal. But if the trouble goes away, suddenly the predator isn't coming in that door, then I don't need to send the signal. But if the signal lingers for too long, then it's out of context with the reason that the signal is being sent. So this becomes really important in neurophysiology because, you know, I need to respond quickly to something. I respond, but if that response lasts too long, the next bit of information that might cause me to have that same response might be polluted like the previous one. So this happens in our bodies all the time. And so there's a place for rapid signaling, and there's a place for not so rapid signaling. Usually in the brain we have rapid signaling. Hormones, on the other hand, and in this case, hormones is referred to chemical messages in the blood, they're usually slower onset, longer duration and responses, so they're more involved with maintaining overall homeostasis. Now there are some hormones that are involved with rapid response. Can anybody give me an example of one of those hormones? Come out of the adrenal gland. Yeah, adrenaline. So, you know, the flight-fright response. Suddenly you're confronted with a predator. You're out in the woods and you see a mountain lion. You need to get away. Okay? And a lot of that response is not just neural, but it's hormone. So the rapid secretion of adrenaline, heart rate goes up, glucose levels go up, energy is mobilized, bang, I can run away. Okay. There are other chemical signals that are a little slower. Can anybody give me an example of a slow, relatively slow signal? Reproductive system. Sorry? Testosterone, yeah, the reproductive system. We don't need to have a rapid response to ovulation. Well, we used to, or maybe we did once. Rabbits do. So rabbits have, they're inductive ovulators. As soon as something goes into the vagina, they ovulate. That's why rabbits breed like rabbits. I'm a reproductive biologist, so I tend to always revert back to what I know. And so, but in us, the menstrual cycle and the male reproductive system are controlled by chemical messages that happen relatively slowly over a lifetime, okay, or over a monthly cycle. So hormones tend to be a little slower, but they also are important for maintaining homeostasis. So your adrenal gland makes cortisol constantly, but it's made in a circadian rhythm. Anybody tell me what cortisol does? Sorry? Stress. What else does it do? It's called glucocorticoid. So it increases the utilization of glucose by the body. So it's not only a stress hormone. In fact, one of the things about cortisol is that every cell in the body responds to it through a glucocorticoid receptor. We're going to talk about that later. So every cell in the body responds to it. If you have too much of it, you die. And if you don't have enough of it, you die. Okay? But we really don't know what it does and how it does it. All we know is you need it. It's one of these stay alive hormones. If you don't have it, you're dead. So cortisol, but so here's another example of a long-acting hormone that is necessary for life and maintaining homeostasis. And sure, it has circadian rhythms. It goes up in the morning just before you wake up, then it goes up a little bit after lunch. But it's maintained in these very strict levels, and every cell in your body responds to it. So it's a slow-acting hormone. And thyroid hormone, you can argue, is similar in that it maintains that metabolic rate. So all these things that are happening in the background of physiology are really controlled by these hormonal chemical signals that are functioning in a long-acting state in order to keep us alive. So these hormones have slower onset, longer duration responses. There's two major subclasses of these hormones. So there's polypeptides, there's protein hormones. These are secreted by a cell that travel to the next cell where they will bind a receptor. And they alter the specific activity of the cells in the of factors in the target cell. And then there's steroid hormones, non-protein hormones, and then there's also the consorts like prostaglandins. And there's actually several other non-protein chemical messages in the body other than steroid hormones. So this list, for the sake of this class, we'll just stick with these two, polypeptide hormones and steroid hormones. Cortisol is a steroid hormone, okay? Testosterone is a steroid hormone. So when, the Bulgarian bodybuilder is on steroids, okay, it's probably the wrong term to use because, some of the steroids that we need to stay alive are not necessarily the anabolic steroids that are used to pump up or to bulk up in the gym. Okay? So steroids alter gene expression primarily, so they have slower onset and a longer duration response, and we'll talk about that in a minute. So you then have local signals, and this is usually occurs between cells immediately. So in a paracrine sense, where you've got a cell communicating with another cell through the extracellular space, extracellular fluid. Great, best example of that, as I said, is the transmission of a signal from an axon, from one nerve cell to another across the synaptic cleft. So here's our paracrine, hormocrine definitions of these locally acting hormones. Okay? So paracrine released by one cell to affect neighbouring cells. Onset and duration varies. And here's some examples of paracrine hormones: prostaglandins. So non-steroidal anti-inflammatories, ibuprofen. How does that work? So one of the main hormones produced when there's an inflammatory response is prostaglandin F2 alpha, a type of prostaglandin. That is part of the inflammatory response locally when you fall on your knee and you get a bruise. That inflammatory response is painful because of the prostaglandins that are being released and the other cytokines as part of that inflammatory response. A lot of that inflammatory response is initiated by these prostaglandins that are made locally. So when you take ibuprofen is a drug that inhibits a key enzyme, cyclooxygenase 2, that's responsible for prostaglandin production. And that then stops this cycle of inflammation, this positive feedback cycle that causes a swelling and pain. And so therefore it breaks the pain cycle and you don't feel pain. Okay? So it's basically inhibiting the production of prostaglandins. You could argue the same for antihistamines. So when you prescribe to your patients later on that they should take prostaglandins, non-steroidal anti-inflammatories for a toothache, now you'll know how those non-steroidal anti-inflammatories are working based on the fact of what I just said about prostaglandin biosynthesis. So prostaglandins, while we're on it, they're also involved with gut motility because prostaglandins are really good at causing, especially the F2 alpha family, prostaglandins are really good at causing smooth muscle to contract. So when a woman is in labour, one of the main hormones, main chemical signals that causes that uterus to contract is prostaglandin F2 alpha. So you want prostaglandins, but you don't want them. So if you inhibit all prostaglandins with something like indomethacin or aspirin to a certain extent, indomethacin is a drug that used to be used because there are two types of enzymes that will make prostaglandins. There's one that's cyclooxygenase 2, that's made in response to inflammation. Okay? There's another COX-1 that's made all the time. All right? So when you're responding to an inflammatory response, when the tissue is in an inflammatory mode, it's COX-2 that you want to inhibit because that's the one that's making the prostaglandins that are specifically doing the inflammatory response. What happens is that if you inhibit both, right, then if you read on the package for non-steroidal anti-inflammatories, of course, they take with a meal and don't take too much because it will cause gut problems. And it causes gut problems because if you inhibit all the prostaglandins, then motility, that smooth muscle of the gut, doesn't quite work right. And then everything clogs up and you're partying all the time. You've got gas and you feel bloated. Okay? So understanding the role of these locally acting hormones and prostaglandins is a great example of one. And the drugs that are used to control their release becomes very important clinically. And then you have autocrine factors that affect behaviour of the cell that releases them. Adenosine is an example, fast onset. There's not a lot of clinical, I can't think of a good clinical example of an autocrine hormone that you can tweak therapeutically that has clinical relevance. But just know that they're there. That's another way of signaling. So how do these signaling molecules alter cell behaviour and effectively alter protein function within the target cell? So the mechanisms can be divided into two groups, and this is based on the way that the hormone gets from one place to another, which is based on its structure. that signaling molecule, that hormone, is either going to be hydrophilic or lipophilic. It's either going to like being in water, it's going to be water soluble, or it's not going to be water soluble, it's going to be fat soluble. So most polypeptides, or all proteins in our body, are generally water soluble. So when the pituitary gland secretes growth hormone, the growth hormone goes outside of the cell and it mixes with the extracellular fluid and then gets transported or goes into the blood system, and it gets taken around through the circulatory system because it is effectively water soluble, and most of what's in us is water. However, when the adrenal gland secretes cortisol, cortisol is a steroid. It does not dissolve in water. So somehow that steroid has to be able to be transported around the body. So how would you do that if you were designing this from scratch? How would you take a lipophilic compound and make it transportable through the blood?
Attach it to an amphipathic protein.
Sorry?
Attach it to an amphipathic protein.
Exactly. So you attach it to another protein. So a lot of these steroid hormones are actually transported through the blood by having them attached to a protein that will dissolve in water. So corticosteroid binding protein is the protein that is present in all of us that transports cortisol around the body. All right? And all of the sex steroids are the same way. They're transported through the body by then binding to other proteins. So, total activity of a hormone is the specific activity times the amount. Okay? And I'm not sure why hydrophilic signals and lipophilic signals are attributed to specific activity and amount respectively, but I think this equation is applicable to whether or not you're a water-soluble or a non-water-soluble hormone. The amount of the signal is always going to be important. Now, I think what is getting at here is that, if you're secreting cortisol, you want it, or testosterone or estradiol, there is a sink of it in the blood because a lot of it is bound by the binding globulins, the binding proteins that are helping this hormone to get through the circulation. And so a change that happens because the adrenal gland is making more cortisol, there'll be a lag time between when that change in concentration actually is sensed by the target cell because of this capacitance of the blood to soak up some of the signal via these binding proteins. And so the amount then becomes affected by the way that the hormone is transported through the blood. And in general, the protein hormones don't usually have to be transported through the circulation. So when growth hormone goes up a little because the pituitary gland is making a little more, it doesn't take long for the circulating levels to go up, and then a cell in the epiphyseal plate or in the liver will respond quite quickly. All right, so let's talk about these water-soluble hydrophilic signals. So these are hormones that are floating around. Because they're water-soluble, they get transported out of the cell through the Golgi apparatus. They usually get packaged into secretory granules and they're transported out of the cell when they're secreted. Okay? These guys cannot get through the plasma membrane because they're not lipophilic. They won't interact with the plasma membrane. They have to be packaged and transported out. Okay? Likewise, they can't get into a cell because they can't get across the plasma membrane. So how are you going to send this signal? Because they're signaling molecules, they're hormones, they have a signal. They want to signal to the cell. How are you going to translate that signal into the target cell? Well, that's done by having the target cell express another protein that it integrates into the plasma membrane called a receptor. Okay, so here's the plasma membrane of the target cell. Here's our hydrophilic signal. It could be insulin, growth hormone, you name it, any protein hormone. It then will interact with and bind to a specific receptor that's sitting on the target cell. And then somehow this binding event needs to be transduced into a signaling event and change the phenotype or the activity of that target cell. And that's really what this whole signaling lecture is all about. How do we get from that chemical messenger actually changing the activity of the target cell? Okay. So, with hydrophilic signals, you've got the hormone binds the receptor, and then what happens is that this receptor then interacts with what's called an effector. Okay? And the effector is usually another protein complex. Sometimes it's the receptor itself, and we'll talk about that in a minute. But the effector is usually another protein complex that's also sitting in the plasma membrane. Okay? And that effectively causes the generation of what's called the second messenger. And this second messenger is the message that is transduced from this signal via the effector to the inside of the cell. So the message from the outside of the cell is transduced to the inside of the cell via the second messenger. And the second messenger is the thing that then goes off and does stuff. Okay? And that stuff can be multiple things. All right? And what it usually does is it phosphorylates protein or dephosphorylates protein. Remember I said phosphorylation is the way that you switch protein activity on or off? Well, second messengers usually initiate a cascade of events that will take protein activity and change that protein activity. And it's this what does the job. Okay? This is what changes the cell's function. Alright, so these signals can circulate in the blood or the interstitial fluid, often rapidly catabolized or metabolized to an activate. And so, here's a context issue again. If your blood glucose level goes up, then you're going to make insulin. That insulin is going to be secreted into the blood. It's going to react with insulin receptors in the liver and the muscle, other places. That then is going to initiate a signaling cascade. The second messenger is going to be made that will switch on glucose transporters that will take some of that glucose out of the blood and package it into, or bring it into the cells, make glycogen in the liver, and so on. Then the circulating levels of insulin will go down, the glucose will go down, so the insulin levels will go down. So there needs to be a turnover of this signal that's somewhat rapid in order for the next bit of signal to have context. So the insulin that's secreted because I just had a meal, okay, is hanging around for too long. When I've finished having that meal and my glucose levels have gone down, then that signal will be out of context. So, as I said earlier, removing the signal is as important as initiating the signal. Okay? So what usually happens is that when the hormone binds the receptor, it initiates the signaling cascade so that the effector triggers the second messenger, but then this whole complex is internalized and degraded. So you end up with a turnover of receptor and hormone together. All right? So that means you need to make more receptor to replenish the sensitivity of the target cell. Okay. So this is actually used clinically in situations like in vitro fertilization, okay? Or other areas of gynecology. Yeah, gynecology, reproductive endocrinology. So if I need to switch off your reproductive system, I can give you lots of a hormone called gonadotropin releasing hormone. We're going to talk about that in a couple of weeks. That's made by the hypothalamus that causes the pituitary to make LH and FSH, which stimulates the gonads in men and women to make sex steroids and gametes. Okay? The reproductive system is controlled by that one hormone. that hormone is produced and secreted in a pulsatile fashion. So a couple of times an hour in all of us, women and men, there is a pulsatile secretion of GnRH from the hypothalamus. Now, depending on the menstrual cycle that you girls are in, it will be, the rate of that pulse will be a little different. But it's pulsatile because when the GnRH binds to the gonadotropin-releasing hormone receptor in the pituitary, it sends a signal, but then the receptor is internalized and it needs time to re-establish it for the cell to make new receptor and put it on the outside of the cell. So commonly what's done is if you have a clinical condition where we don't want these hormones to be made by the pituitary. For instance, if you have prostate cancer, we want to shut the pituitary down so there's no testosterone being made. Or you have polycystic ovary disease, we want to decrease the level of the activity of these hormones to get that ovary back in control. Okay? So what you can do is give a woman a long-acting and a lot of gonadotropin-releasing hormone. Instead of it being pulsatile, it would be constant. And because it's constant, it downregulates the receptor so much that the pituitary is just overwhelmed with all this signal and just completely shuts down. And so then the whole reproductive system shuts down. It's counterintuitive. You think if you give more GnRH, you actually will stimulate the reproductive system. But it has to be secreted in a pulsatile manner because of this concept of signal being in context and in synchrony with the target cell. Okay? Insulin is different. Insulin, that whole system, there is a surge of insulin immediately after a meal, okay, but then it sort of wobbles and plateaus out and you end up back into metabolic homeostasis, okay? But, just keep in mind that these systems have evolved to be finely tuned so that the hormone and the receptor know what they're doing. The target cell has evolved in order to respond appropriately. So internalizing the receptor is an important way of regulating the signal. So what's here then? The signal can circulate in the blood, you went through that. The receptor is no good without the effector. It is specific for a particular signal. Right, so that's where the specificity comes in. It's a lock and key type mechanism. The lock is the receptor and the key is the hormone. Right? And the two have to match. Sometimes there's a slight mismatch, and you can take advantage of that therapeutically if you understand the biochemistry of this hormone receptor interaction. You can tweak the hormone slightly so that it fits the lock in a different way that causes a different response. Getting back to our gonadotropin-releasing hormone example, the reproductive system, this hormone is made by the hypothalamus. We wouldn't be here if it wasn't for this hormone. Okay? It's what regulates reproduction. Gonadotropin-releasing hormone is only 10 amino acids. It's really small. And all it has to do is get from the hypothalamus to the pituitary. That's all it has to do. So it goes through the portal system, gets to the anterior pituitary, does its thing. Now, because it's so small, what the drug companies have been able to do is tweak it. So they might take that tyrosine and change it to a serine, that glutamine and change it to a threonine, and then look to see what effect that has on the protein. And some of these analogues are, it turns out, long-acting and can be used therapeutically. Because the protein is so short, it lends itself to being a druggable target. And so there are other, insulin is a little bit more difficult. You saw the structure of insulin last week. It's sort of a disulfide bond, it's a proinsulin. That's a little bit more difficult to tweak, but there are insulin forms, glargine, insulin glargine, where they've tweaked the molecule so it sits on the receptor a little longer and its half-life in the blood is a little longer. Okay? So by understanding the structure of the hormone and the structure of the receptor, then you can start really going to town with that and playing around with, well, how can we stop this particular disease, given that we know that it's regulated by this hormone-receptor interaction? Likewise, you can, by understanding the effector and the second messenger, that's another drug to target. Okay? So by understanding these pathways, it's not, we're not just, we're not just studying this for, well, but in basic science we do stuff because it's interesting, but it has clinical relevance. It has relevance to the biology and to clinical practice. All right, so let's talk a little bit about lipophilic signals. These are hormones that are flowing through the system that do not solubilize in water. Okay, so what's nice about them is, well, not nice, but what's unique about them is they can, because they're lipophilic, they pass straight through the plasma membrane. So these hormones can get into a cell very easily. Okay? And typically these are steroid hormones. Cortisol, testosterone, estradiol, progesterone, and there's a bunch of others. But they're the main ones. So they can pass through the membrane, so they can't be synthesized ahead of time and stored in vesicles. And this is what usually happens with protein hormones, that you have insulin that is stored in vesicles within the cell, and then when the glucose levels change and the pancreatic beta cell needs to release insulin, it first releases those stores and at the same time makes more to replenish the stores, and so you have this ready-made amount of insulin ready to be released. Steroid hormones is different. With steroid hormones, you have to actually synthesize the hormone every time. Okay? And in that case, what happens is the cell takes in cholesterol, or it makes its own cholesterol, and converts it through the steroidogenic pathway. And I won't put that up, I'm glad he didn't. It's basically 2-deoxy chicken wire all over the place. So there's the structures, you don't need to understand the structures of steroids. Well, you do if you're a regular endocrinologist. As dentists, I don't think you necessarily need to. But what you need to understand is that these hormones are made by the cell immediately in response to a signal that's telling them to make hormone. So what can anybody think of a signal? I just talked about signals from the brain that tell other organs to make hormones. One of them is the adrenal gland. Let's stay with cortisol. So cortisol is made by the adrenal gland, the adrenocortical cells, and it's made in response to adrenocorticotropic hormone, ACTH, produced by the anterior pituitary. And that is made in response to corticotropin releasing factor, or hormone, that's made by the hypothalamus. So you have hypothalamus telling the pituitary to make ACTH. The ACTH then acts on one of these receptors, because ACTH is a protein, okay? It acts on the ACTH receptor on a target cell within the adrenal gland, okay? It gets activated because of that binding. That binding then stimulates an effector and a second messenger. That second messenger then switches on the expression of genes that encode for enzymes that make steroids. And some of those enzymes are required to make cortisol. So you make more of those enzymes, and now bang, cortisol goes up. Right? So in order for cortisol, the steroid hormone level to go up, you actually have to make more of it, de novo, by the target cell. And it's all a matter of increasing the amount and activity in certain cases of these what's called steroidogenic enzymes within the target cell. Okay, so you have let's now think of this cell as being the adrenal cortical cell that is making cortisol. It's making, the stimulus comes in, and that's the ACTH comes in, tells the adrenal cortical cell to make more cortisol. It does that by telling the cell to make the enzymes that are making the cortisol. The cortisol then gets released, goes straight through the plasma membrane. It then binds, or it actually gets into the circulation very quickly, but when it's in the circulation, it'll bind to a transport protein. sometimes albumin, can be other specific proteins, sex steroid binding globulin, corticotropin binding globulin. In any event, it gets transported through the blood. Now, there's always this balance between free and bound. Okay? This is a dynamic situation. There's always a little bit of free around, even though it's not water soluble. And that little bit of free, even though that most of it is bound, can actually diffuse out and occupy this extracellular space and diffuse into a target cell. All right? Cortisol goes up, goes into the blood. There's a little bit of free around, so the level of cortisol in every interstitial space in the body then goes up. Now, if it happens, then that cortisol will go into that target cell. If it goes into this cell as well, it goes into all cells because it can go through the plasma membrane. But it will only have effects, just like the insulin will only have effects on cells that are expressing the insulin receptor. The steroid hormone, whether it be cortisol, progesterone, testosterone, estradiol, will only have effects if that target cell expresses the steroid hormone receptor. Okay? So now we've got a target cell that knows it wants to respond to cortisol because it's expressing the receptor. All right? There's a gene that encodes for the receptor, it's being expressed, the cell's got the receptor. And what the definition of the receptor is that it's a protein that has a high affinity for the hormone. So it will bind the hormone. And usually what happens is that the steroid binds to a specific part of the receptor called the ligand binding pocket, or hormone binding pocket, and it sits in there really tightly. Okay? But once it gets in there, it causes this protein to actually change its function, just like the circulating protein hormone will bind to the cell surface receptor and cause it to change its function. So the receptor becomes activated by binding of the ligand. Now in this case what happens is that these receptors are what's called ligand-activated transcription factors. And they're usually in the cytoplasm, but they can also be in the nucleus, just hanging around free. But when the hormone is around, when the steroid comes along and binds the receptor, then it activates those molecules to become transcription factors. And what transcription factors are is proteins that will either switch on or off gene expression. So they become transcription factors. They will bind to DNA in the promoters of genes that have response elements. So here's another signaling molecule receptor type situation where there's a sequence of DNA to which this activated receptor will want to bind. So it'll sit on it and bind there. And when it binds there, it's usually in the promoter sequence of genes. This is the front of a gene that's the switching component of the gene. It tells the gene, yes, be expressed, or no, don't be expressed. So it binds to that area and then affects the transcriptional apparatus, and you will get then a change in the expression level of that gene. When that happens, the mRNA level changes, that changes the level of protein, and it's this protein that changes the function of the cell. So here we have an indirect mechanism of changing the response of the cell via the production of new proteins. Okay? And so that all comes down to, as he says here, we'll talk about gene expression and regulation soon. Won't that be fun? But it all comes down to regulating the amount of protein within the target cell and the activity of that protein. So, getting back now to the hydrophilic signals. So that's these guys here. And let's talk in a little bit more detail about these second messages. Because these are really what cause the cell to respond in a specific way. And they're really nice experiments that were done 20 or 30 years ago, where people took components. So what happens is that as I said, the hormone has to interact with the receptor. The receptor then has to interact with or activate an effector. And the effector then creates a second messenger that actually does stuff. Right? And you can tweak this by rearranging the connection between the receptor and the effector. And this is the way people actually began to understand these molecules, by using molecular biology to actually change the structure of some of the receptors. So, and I won't go into that, it's a little complicated, but let's get back to this. So the receptor recognizes the extracellular signal. The effectors are stimulated or inhibited by receptors and produce intracellular second messages. I've said that 3 times now. There are 3 types of receptor-effector relationships. So receptor and effector are the same protein, and that happens with steroid hormones. Okay? So ligand binds the steroid hormone receptor, it then becomes activated to be a transcription factor. So it becomes the effector once the ligand is bound. And also with some protein hormones, when insulin binds the insulin receptor itself becomes what's called a tyrosine kinase, which is an enzyme that phosphorylates other proteins. And so the receptor itself gets activated to become the effector. And so receptor and effector are subunits so this is the other type, where the receptor and effector are subunits in the same protein complex. So it's like the acetylcholine receptor. So in this example, you have hormone binding the receptor, and the receptor then binds the effector, activates the effector, and then it can do its thing. And the other class is that the receptor and effector are separate proteins. And this is the most common type and requires a way of communication between the receptor and the effector. So in this situation, the receptor is sitting in the plasma membrane, and so too is the effector. And once the receptor is activated, then through another second messenger, the signal is transmitted to increase the activity of the effector, and then it can then go and do its thing. So neurotransmitters, things like ion channels and membrane proteins. So how do they signal? So these are acetylcholine, noradrenaline, the GABA receptors, glutamate. These are usually small molecules, okay, that are acting as at the synaptic cleft as neurotransmitters. So what these guys do is that they usually change the activity of ion channels within the target cell. So have you guys done any electrophysiology yet? Potassium, sodium gradients across the cell? Good. So what these guys will do is they'll either activate a sodium influx channel or a potassium outflux channel. But effectively, the neurotransmitter binds the receptor, and the receptor then is directly linked to or affects the gating of an ion channel to affect the electrical activity of the cell. All right? So, and this is really quick. And then this usually will set off an action potential that will transmit down the neuron. So there are other situations where, and let's go back here. Usually these guys are linked to either sodium influx or potassium outflow in order to depolarize the membrane. Okay? And that will cause an action potential. But in other cases, the receptor is linked to a calcium channel. Calcium becomes a really important second messenger. We need it to actually contract smooth muscle, and we need it to regulate heart rate. Okay? So it's important in smooth muscle, and it's important in a lot of other cell types as a second messenger. So you have a hormone interacting with the receptor. That is linked to a calcium channel. That causes calcium to be transported into the cell. So now the intracellular levels of calcium are going up. And if you look at the gradient here, it's 2 millimolar outside the cell and 2 micromolar inside the cell. So there's 1,000 times more calcium ions on the outside of the cell than on the inside of the cell. If you open this channel, all of a sudden they're just going to be flowing down their concentration gradient and into the cell. When these calcium ions get into the cell, they bind with a calcium binding protein called calmodulin, and that becomes an active complex that will interact with other proteins and change cell behavior. All right? So this is a signaling pathway that happens in a lot of smooth muscle, causes smooth muscle contraction, heart muscle, skeletal muscle as well, and other parts of the body. But this it has to be inactivated. So this, again, switching off the signal is as important as switching on the signal. All right? The signal comes off the receptor and no more calcium enters the cell. So that's when the hormone receptor complex gets internalized, the signal gets switched off, more receptor gets made. If there's more hormone around it, it will repeat the same thing. But there's a level of switching off here, but there's also a level of switching off downstream where intracellular calcium has actually begun to be pumped outside the cell in order to get things back to normal. All right, so this is a bit of biochemistry here, but it becomes important in many physiological situations. So calcium that did not enter is pumped out of the cell or into the mitochondria, or it's stored in the endoplasmic reticulum and cytoplasmic reticulum. So the consequence is that intracellular free calcium drops and everything comes back to normal. Now, I talked about tyrosine kinase receptors, and the typical one here is the insulin receptor. So when insulin binds its receptor, it will cause a conformational change in the receptor and activate this tyrosine kinase. And kinase is the name of an enzyme that phosphorylates another protein. And tyrosine kinase will phosphorylate at tyrosines. Okay, so the signal will come in. It'll bind the receptor then will become activated, the tyrosine kinase activity will be activated, and then that causes an activation of the receptor. It generates energy by converting ATP to ADP to phosphorylate the protein, and then that phosphorylation switches on the effector activity of the receptor. All right, so this also has to be switched off. Okay, so there are other proteins called phosphatases that will take that phosphate and turn it off, and then the receptor can be recycled back into being in the active form. You also want to get rid of the ligand, of course, because if it sits there, you've got no place for another ligand, for another signal to come and do its thing. So usually it gets phosphorylated, this whole thing gets internalized, and a new receptor gets internalized and degraded. All right. Some of the proteins that are phosphorylated by tyrosine kinase are themselves protein kinases. So this is a cascading effect and an amplifying effect. So by binding one receptor, you could phosphorylate multiple tyrosine kinase molecules or other protein kinase molecules, and that amplifies and the signal within the cell. Human cells have almost 2,000 different protein kinases, 1,000 different phosphatases. So phosphatases dephosphorylate, kinases phosphorylate. So basically what's saying is that the receptor is actually acting as its own effector by increasing that kinase activity in response to ligand binding. All right, now the other large group of receptors that are important are what's called G protein receptors. And they're called G protein receptors because they're proteins in the cell membrane. And you may have seen images of this. They're also called serpentine proteins because they snake in and out of the plasma membrane with an extracellular and intracellular domains. And they have 7 transmembrane crossings that happen in this long molecule. But what they are is, first of all, they're receptors. They're extracellular dangling bit on the outside will actually bind a hormone, glucagon or epinephrine or ACTH or growth hormone. But then what happens is that once that happens, it causes a conformational change of the receptor, and then it then activates a G protein, a GTPase, a protein that will take GTP and turn it into GDP, that will then activate an effector. Now, the best example of this is the link between G proteins and this enzyme called adenylyl cyclase that causes the production of cyclo-AMP. Okay, so this is a very common signaling pathway within our cells. And we tweak it a lot because we drink caffeine. And caffeine, coffee, anything that has caffeine in it will alter our cyclo-AMP levels. And that's why we get caffeine buzz. And we'll talk about that in a minute. So many systems involving different signals and receptors are acted by cyclo-AMP, glycogen metabolism. So cyclo-AMP is a very common second messenger. So let's have a look at this. So the hormone binds the receptor. The receptor is a G protein coupled receptor. So it then will make GTP, which then activates a protein called adenylyl cyclase. It will take ATP and change it to cyclo-AMP. Then that cyclo-AMP will then, within the cell, go off and do other things. And it's those other things that cause the change. So the G protein it's called the G protein because it binds GTP1, glutamine triphosphate. They're usually in the membranes, and they're activated by the binding of the receptor. They diffuse in the membrane. Within the membrane, they can sort of slide across and activate the adenylyl cyclase that's also in the membrane. And these receptors there's a sort of a complex of proteins, 3 different proteins, the alpha, beta, and the gamma complex that interact with the receptor. And so what happens is, in the resting state, alpha, beta, and gamma are sitting in the plasma membrane, so too is the receptor. Nobody's talking to each other, all right? There's no hormone around, so there's no reason for these guys to interact. That's the resting state. And this alpha unit is bound to GDP, and it's GTP that we want, not DP. So, hormone binds, it causes the receptor to alter its configuration, so now some dangling bits here, and it interacts with this alpha, beta, gamma complex, and that activates the GTPase activity of the alpha complex, such that it will take, this is wrong, it usually generates GTP. Okay? That GTP, right. So it's able to generate GTP. So now you have an alpha beta complex that has GTP associated with it instead of GDP. In this state, it is activated. It can then go and interact with another effector molecule, and in this case it's the adenylyl cyclase enzyme that causes the generation of cyclo-AMP. But in doing so, when it interacts with this effector, this adenylyl cyclase, it causes the alpha-beta subunits to fall off, and then that inactivates. So it gets self-inactivated by virtue of the fact that it binds the effector. Okay? And then this whole thing can recycle back to form the alpha, beta, gamma in the GTP state. So this is a way of recycling this intermediary alpha, beta, gamma complex in order for this signaling to keep going. And there are certain things like chlorotoxin that inactivates GTPase activity, so if that's the case, these things will all shut down. That's what causes diarrhea and potential death. It's interesting that this whole system was discovered by Alfred Gilliam, and he was an alumnus of Case, who won the Nobel Prize in 1969. So there's a whole family of these alpha, beta, gamma complexes that link with specific receptors. And some of them will increase adenylyl cyclase activity, and some of them will decrease adenylyl cyclase activity. So effectively, you get to a situation where you're regulating the level of cyclo-AMP within the cell, and that's that second messenger that's critical. So what does cyclo-AMP do? All right. So let's say we've activated the adenylyl cyclase because of the activation of this GTP G protein complex. takes ATP and converts it to cyclic AMP. The cyclic AMP then can bind to other proteins and activate their kinase activity. And it's a common theme with this whole signaling lecture is that proteins get changed by phosphorylation. So phosphorylation within the cell is a very important switching mechanism. So just as with other signaling molecules, you want to get rid of the cyclic AMP as well as make the cyclic AMP, or else it'll accumulate and keep having its effect. Okay? And the thing that gets rid of the cyclic AMP is an enzyme called phosphodiesterase. There's lots of these, and they're constantly working in the cells. They're always chugging away in the background, keeping this under control. All right? When we have theophylline or caffeine, it inhibits the activity of the phosphodiesterase. So caffeine is a phosphodiesterase inhibitor. That means the cyclic AMP levels are going to pile up in our cells, and this signaling pathway is going to be amplified. Okay? So that's how we get the morning coffee high. All right. Now, it's only two more pathways to go, and then we're going to call it a day. So phospholipase C. So this is another pathway through which these G protein coupled receptors will act. So again, we have a receptor on the plasma membrane. Ligand binds, it activates a G protein coupled complex, and that activates an effector. And in this case, the effector is an enzyme called phospholipase C. So what usually happens is that within the plasma membrane, there's these things called phosphatidylinositol diphosphate, PIP2. Okay? What phospholipase C does is it takes PIP2 and converts it to a thing called diacylglycerol. And it's this diacylglycerol, which is like the cyclic AMP of the previous example. That becomes the second messenger. Right? So if you activate this phospholipase enzyme, it converts the phosphatidylinositol diphosphate into diacylglycerol and inositol triphosphate. So two things. Diacylglycerol and inositol triphosphate. These guys then become the second messengers that go and do stuff in the cell. Alright, so diacylglycerol is hydrophobic and remains in the cell membrane, whereas IP3 is hydrophilic and can enter the cytosol. Very complex piece of work this, but it's not that hard if you break it down. So here's our IP2s, PIP2s get made. Here's our receptor. The protein binds the receptor, phospholipase C gets activated, now it makes diacylglycerol that stays in the plasma membrane, and it makes IP3 that can go into the cytoplasm. The diacylglycerol actually activates a protein called protein kinase C. And then this then can initiate a kinase cascade that'll phosphorylate other proteins that'll cause a change in the effect or the function of the cell. IP3, on the other hand, goes to the endoplasmic reticulum. Remember I said calcium can be stored in the endoplasmic reticulum, and it causes the release of calcium from the endoplasmic reticulum, and now you activate this calcium cascade. So you're activating two types of signaling cascades, a protein kinase cascade, but also the calcium cascade. So calcium binds calmodulin, and calmodulin will then bind a protein kinase. It all comes down to protein kinases. Then that'll change function. All right? So this is another major pathway in the second messenger pathway that's common to many hormonal systems. Okay? So why is it that if there is, it's always bothered me that, you know, ACTH, GnRH, all these different hormones, FSH, will all signal via cyclic AMP. Okay? So if they're all signaling via cyclic AMP, what does it matter that they're different receptors, they're all going to have the same effect. Well, what happens is that the cyclic AMP, these second messengers within the cell, aren't just swimming around. It's not just a bag of water. There's actually other systems that compartmentalize them to different biochemical pathways. So if you're a granulosa cell sitting in the ovary and FSH hits the cell, yeah, sure, you'll get cyclic AMP made in that cell. But because you're a granulosa cell, you're responding, you have other things going on in there that make you a granulosa cell, so you'll respond to cyclic AMP very differently than if you're an adrenal cortical cell in the adrenal responding to ACTH, even though it's cyclic AMP that's doing the same thing. So keep that in mind, that it's cell context dependent. Now, one of the other things that happens in this pathway is that in order to make the PIP2, it releases what's called arachidonic acid. And arachidonic acid is a substrate for prostaglandin synthesis. So this one pathway is activating a protein kinase cascade, calcium signaling, and also potentially increasing the secretion of prostaglandins from these cells. So this is a really important pathway when it comes to inflammation and pain. Okay? All right, so the last one, really interesting one, and this is an old lecture that was probably put together in the early 2000s by this last slide, because that's when nitric oxide was discovered as a chemical messenger. So we all think of nitric oxide as being a gas, and it actually is. It's not nitrous oxide, that's what that's the anesthetic, or analgesia. It's nitric oxide, NO instead of N2O. So the way this was discovered was that we knew that if you think about a blood vessel, okay, a blood vessel, especially an arteriole, has smooth muscle around it, and it's got endothelial cells lining the inside. So these are the endothelial cells, are the main cells that the blood is in contact with. But around the endothelial cells is a layer of smooth muscle. So when blood pressure changes in your body, it can change because those smooth muscles around the arterioles are contracting. And if they contract, they'll decrease the diameter of the arteriole. And if you decrease diameter, you can increase pressure. It's just like opening a faucet. And if there's there's lab faucets where they have this little conical spigot at the end, all this would come out really fast. Right? But it's the same amount of water, it's just that you've decreased the diameter so the pressure increases because the same amount of water is coming out. All right? So blood pressure is controlled by the, in many instances, by the tone or the contractility of the smooth muscle cells that are surrounding these arterioles. Okay? But it wasn't understood how the contractility of those vascular smooth muscle cells is actually controlled. And it turns out the endothelial cell is needed to relax the smooth muscle cells. So that this endothelial cell, this first cell on the inside lining of the blood vessel, actually makes something that causes the smooth muscle cells to relax and open up. And that's how blood pressure goes down. Okay, so more blood can get to that organ. Right? So smooth muscle or vascular relaxation is mediated by a second messenger or hormone that's being made by the endothelial cells. So people were taking this apart. They were isolating endothelial cells, they were isolating the smooth muscle cell. They said, what the hell is this endothelial cell making that's causing the smooth muscle cell to relax? A lot of work, a lot of years. Eventually it turns out that they found that it was nitric oxide. So, and it was originally called endothelium-derived relaxing factor. So they knew that these endothelial cells were making something that was causing vasodilation. It is a very, very unstable free radical that exists for 6 to 10 seconds before being disintegrated. So again, this is a really rapidly acting hormone. chemicals messenger that you don't necessarily want it to act for a long time. You want to relax, and you want your vascular system to respond to the environment that it's in and the physiological systems that are telling it to either increase or decrease blood pressure. It's also implicated in preventing platelet aggregation, and it's a neurotransmitter. So as it turns out, it was discovered in the vascular system, but it's now known to affect lots of other systems. So how does this work? So in one cell, what happens is that cell expresses a thing called nitric oxide synthase, which is an enzyme that will take arginine and convert it to citrulline, and in the process, it combines oxygen with nitrogen, and you end up with NO. The NO, because it's a water-soluble gas, will literally diffuse out of the cell to a neighbouring cell, and it will activate guanidinylate cyclase. Remember I mentioned adenylate cyclase that makes cyclic AMP? So guanylate cyclase will take cyclic GTP and convert it to cyclic GMP. This then becomes the second messenger. All right? Okay? So this is very important in vascular relaxation. I think that's the last slide here. So let's go back to that. I should have done that. Where's the definition? There it is. So, a couple of other little... Let's go again. So, nitric oxide activates guanidinylate cyclase and it causes relaxation. Let's just go to this last one. All right. Now the interesting thing about this is that this guanidinylate cyclase becomes a really important second messenger for smooth muscle, vascular smooth muscle relaxation. So can anybody tell me a well-known, very popular drug that is an activator of guanidinylate cyclase? You see ads for them on TV all the time. Viagra, exactly. So if you relax the smooth muscles that are supplying blood to the penis, you're going to cause an erection. Okay? So that's how the guanidinylate cyclase. So they were working on blood pressure. They were working on drugs that you could give to people that have hypertension that will decrease their blood flow. And it turned out that when they were playing around with these guanidinylate cyclase activators, they weren't that good at changing blood pressure, but they were good at giving guys a whip. So they moved from that to where the market is. The other thing about it is that this is nitric oxide, and nitric oxide is also released. What's another way that you can make nitric oxide for people? Some people take nitroglycerin, okay, who want to relax the angina that they're feeling because of the blood vessels that are supplying blood to the ventricles are a little constricted. Okay, so they'll take a nitroglycerin tablet, put it under their tongue. It increases NO levels throughout the body, but also in those blood vessels that are supplying the heart. So nitroglycerin is an explosive. It's always bothered me a bit of that. It turns out that people that worked in the dynamite industry knew this a long time ago. They were handling all these nitrous, this, that, and the other thing, and were increasing their NO levels and going home at night with an erection and a very bad headache because their blood pressure would be low. So anyhow, this is a common thing in the dynamite industry, that they would, put a bit of whatever it was in their pocket and take it home at night so that they could, that was their form of Viagra. So the nitric oxide story is very interesting. All right, so we're done with this slide deck. Is there any questions? Anything you didn't get? Anything you feel you don't need to get? Yes?
So which cell creates the nitric oxide from the arch?
The endothelial cell.
So the vascular cell that's written right next to it, in which the nitric oxide passes through. Is that also an epidural? So is that
No,
the smooth
this
muscle?
this should be a vascular smooth muscle cell.
Smooth muscle.
This is the endothelial cell right here. Good. If you have any questions, contact me. I should have put my, you should have my email somewhere on Canvas, but I'll put it up now. I don't use PowerPoint, so I have no idea what's going on. Anyway, it's SXM188. And it's Messiano, M-E-S-I-A-N-O, first name Sam. All right? So that's self-signaling. We're probably never going to come across this again. It's handy to know. You now know how ibuprofen works. You go. We're done. Or not.
Just go the opposite.